Okay, everyone. Thank you so much. And I think dovetailing off of the last session, we need more targets, and particularly in the immunotherapy space. And so we're lucky to have an incredible panel of people who are all brilliant and going to tell us about their work. And so the first person is Dr. Avery Posey from Penn. I actually had the good fortune of meeting Dr. Posey when I was a fellow, and there was a forum for young immuno-oncology interested people, and he was one of the mentors. And I remember just being blown away by his work, but also his mentorship abilities and general kindness as well. And so today we're going to hear about some of his exciting work in CAR T cells. He doesn't focus primarily on kidney cancer, and I think we'll provide a what? No, we're not, but is one of the thought leaders in CAR T cells, and we're lucky to have him here today. Thank you.
Oh, sorry. So we're not going to hear about my work.
Oh, okay.
I'm going to talk about CAR T cells in kidney cancer and what we've learned in the last couple decades. So a CAR is I'm sure this is not new to this group. It's a TCR-like molecule that combines antigen binding properties of a monoclonal antibody in its most conventional format to the intracellular signaling domain from immune activation receptor like the TCR, CD3 zeta, or at gamma chain of Fc receptor. And that's the most conventional format. But what might be relevant to this crowd is CD70s. It seems to be a promising target in kidney cancer. And there are other formats of CARs that are developed that use antigen binding partners, such as CD27 as the extracellular portion for the CAR molecule itself. And in the early 90s, when these molecules were developed, this was the format. There was no co-stimulation that was added later in the early 2000s. And these second generation CARs that add CD28 or 41BB based co-stimulation are all are the formats of the 6 FDA approved CAR T cells that target B-cell malignancies and multiple myeloma. But the earliest renal cell carcinoma trial in kidney trial for CAR T cells was in the Netherlands, targeting CA9. And you don't have to be convinced that CA9 is a promising target in kidney cancer for immunotherapy, but there are limitations for CAR T cells that go beyond the limitations of other antibody-based therapeutics, and namely antigen expression in normal tissue. So starting in March 2003, this trial initiated with 3 separate cohorts using a first generation CAR. In the first cohort, it was a dose escalation. Second cohort was straight dose of 2 billion CAR T cells given in 2 cycles over 5 days each cycle. And there was toxicity observed in the first 2 cohorts, high levels of liver enzymes in the sera, suggesting that there might be some on-target toxicity. CA9 seems to be expressed in the bile ducts of the liver, and immunohistochemistry of CD4, CD8, and CD3 for T cells suggested that there might be some CAR T cell activity, although due to technical limitations, they weren't able to show you that these were CAR T cells. And so in the third cohort, pre-treatment of patients with an anti-CA9 antibody, the same antibody that the variable domains of the CAR are derived from, was used, and that decreased the toxicity. And so this was the first example. It was one of the first examples of on-target off-tumor toxicity with CAR T cells, but it was also the first example of a workaround, a way to overcome that limitation. There were no clinical responses noted, but this suggested that the target needs to be considered more carefully than other antibody-based therapeutics. Notably, this was a first-generation CAR. The CAR T cells were generated through transduction by a gamma retrovirus. We now know that gamma retroviruses driving transgene off the LTR are susceptible to silencing. And so there was a decrease in circulating CAR T cells, both identified by flow cytometry, peripheral blood, as well as a ratio of CAR transgene RNA to incorporated DNA. In addition to silencing of the receptor, there was also a immune response that patients developed against the CA9 receptor. The variable domains are derived from a mouse antibody. And so patients develop human anti-CAR antibodies, as well as a cellular response, so T cells that were reactive against these CAR T cells to deplete them. Importantly, the pretreatment with the anti-CA9 antibody did suggest that helped increase the persistence of these CAR T cells. And so that may be a consideration. This was a first generation CAR, so second generation molecules may have better activity in this case. And there are a number of CAR T cell trials in kidney cancer that are ongoing. Some that are in China targeting ROR2 or AXL, another targeting CA9, that is a second generation CAR, but three that I am going to talk about are VEGF receptor and then two CD70 CARs. So the VEGF receptor CAR trial was done at NCI. This was a CAR that was developed from in Steve Rosenberg's group. First, they demonstrated proof of concept using an anti-mouse VEGF receptor CAR to show that they could inhibit the growth of these established vascularized syngeneic tumors and enhance survival of mice. And then in the same work, they developed human CAR T cells and showed in vitro that these CAR T cells were lytic. Did not model this in mice. There may be limitations of modeling human VEGF targeting CAR T cells in mice without a human VEGF receptor knock-in, and likely with ligand as well. This trial proceeded starting in October of 2010. It was a third generation CAR molecule, also transduced with a gamma retroviral, and I told you the limitations of that method of transgene expression. Patients in this case received lymphodepletion. The prior study, the CA9 study, there was no lymphodepletion. We know that lymphodepletion is necessary for the engraftment and persistence of these CAR T cells. It definitely improves their activity. These patients also received up to 15 doses of high dose IL-2. And it was a dose escalation study of 11 different cohorts, but there were no objective responses observed. We don't have correlative data on these that's reported from this trial, so we don't know the persistence of these T cells. But most recently, a group in Lausanne and George Koukos's group demonstrated that VEGF binding to VEGF receptor actually can inhibit the activity of these CAR T cells. And so using bevacizumab in combination with these CAR T cells may be a way to overcome the lack of activity for this CAR. The other two trials that I think are interesting are both targeting CD70, and we've had lots of discussion about CD70 yesterday and today. CD70 is highly expressed in renal cell carcinoma. It does have some low-level expression, particularly in lymphocytes, and it can be modeled in cell lines. And it's expressed in different intensities across multiple subsets of renal cell carcinoma. And so one of the limitations for CAR T cells is that the on-target off-tumor toxicity can be severe and it can lead to lethality. We've seen this with HER2, mesothelin, and other CAR targets. But an approach to overcome this limitation is to use that differential expression in which normal tissue has low-level expression. If you alter the affinity of the scFv that is used to target the target antigen, you may be able to avoid targeting low antigen expressing cells. And so that's one approach that can be optimized in this area, and depends on your binder. As mentioned, lymphocytes express CD70 and mostly activated lymphocytes. So fratricide is a possibility. In this work, Allogene developed several different binders to CD70, characterized those into two different classes based on the epitopes that they bind to, and identified binders that would mask the expression, the self-expression of CD70. And so these are class 1 CARs that when expressed in the T cells the CAR would express, but CD70 would not be expressed or be able to be identified by a particular immunoflow cytometry reagent. Whereas class 2 CARs would co-express CAR and CD70. This can likely lead to constitutive activation, antigen stimulation in your CARs, and exhaustion. in terms of in vitro prediction of on-target toxicity, which is an area that we've added on in CAR T-cell development, preclinical development, the binder, in this case, a CAR23 binder, which has been used in the Allo316 product, was used, just the binder was used for immunohistochemistry, demonstrated reactivity against multiple cell lines, no reactivity against a knockout cell line, important, but did show some surface expression on the lymph nodes, on lymphocytes, and maybe some cytoplasm expression in kidney. And so in order to evaluate whether there's a potential for on-target off-tumor toxicity, now we can use primary cells from the kidney in cortex or medulla and do co-cultures. So first immunohistochemistry, but also co-cultures of these CAR T cells and look for lytic activity. In this case, Allo did not observe this. And so this product is generated through use of allogeneic T cells. These cells are off the shelf. They are transduced with a lentiviral vector, and then talons are used to genetically edit both the TCR alpha chain, and CD52. The use of CD52 knockout allows combination treatment with an anti-CD52 depleting antibody, and that should increase the persistence, the engraftment persistence of these CAR T cells. Notably, the manufacturing process for these cells is longer than we seem to start to prefer in CAR T cell research. We know that early manufacturing or shortened manufacturing of these cells preserves a central memory phenotype that correlates with better responses. There's a short decrease in viability that recovers after the TALEN gene editing, and then cells can be purified based on loss of TCR, and that's meant to prevent graft versus host disease. This trial is a dose escalation that's going from 40 million to 120 million CAR T cells, and the patients are lymphodepleted. And reported at AACR this year, there are clinical responses, and so in both a CRISPR Therapeutics trial and in this allogene trial, both targeting CD70, we're starting to see clinical responses in renal cell carcinoma with CAR T cells, which is encouraging. And so in this case, these are 3 patients with a partial response, 2 confirmed, 1 unconfirmed, and notably they're at the lower dose levels. One of the benefits of CD70 being expressed on endogenous T cells is that these cells, similar to maybe CD19 CAR T cells, are going to receive some constant stimulation, so it may enhance their persistence and activity in patients. And then the CTX130 CAR T cell trial is a CRISPR edited trial using Cas9 to knock out beta-2 microglobulin to remove MHC class 1, the track TCR alpha chain to remove the TCR, and a knock-in of the CAR into the TCR alpha chain, constant alpha chain locus, and the knock-in of a CAR into the endogenous TCR locus has shown better expression of the CAR molecule, more stable expression compared to a retroviral or lentiviral transduction. And this CAR is then regulated in the same way that a TCR will be regulated. So upon antigen stimulation, the CAR is downregulated and then re-expressed after the cells has time for rest. This seems to preserve T cell activity because those T cells will avoid chronic antigen stimulation. In a trial of cutaneous T cell lymphoma, this CAR has shown activity. And CTX131 is a new trial from CRISPR Therapeutics that adds to the CTX130 anti-CD70 CAR by knocking out 2 additional molecules, regnase-1, which is a negative regulator of many T cell cytokine functions, and TGF-beta receptor 2, which allows suppression of T cells through binding of TGF-beta. And so in preclinical work, they've shown that these CAR T cells can overcome some of the limitations of CTX130, which is shown here in black, and so by increasing potency. So this is a CAR T cell trial that is still virally mediated for transduction. The CAR is introduced through a HDR homology-directed donor through AAV. There's no gamma retroviral or lentiviral vector used here. The T cells are edited for TCR, MHC class 1, and regnase 1 and TGF-beta. And in manufacturing these cells, these cells show increased central memory frequencies when compared to CTX130. And so this is the phase 1 trial that's currently enrolling. So I didn't talk to you about my work because we don't do my lab is very tumor agnostic. We focus in many different solid tumors, focusing on a class of antigens that we think are more tumor specific because of a change in post-translational modifications. But I thought more relevant to this work is where are we advancing in this field. And so the initial CAR T-cell trial 20 years ago had several limitations that we've learned from now in the field. We know that we need at least a second generation CAR molecule, an alternative to gamma retrovirals, either viral or non-viral, humanizing the variable domains of the CAR molecule, lymphodepleting is necessary, avoiding expression in normal tissue, identifying new antigens, targets are important. And then one thing that none of this work has tackled yet is we must target multiple antigens. And the same is true for the small molecules in the talk we heard earlier today. Targeting one single antigen is just going to allow escape of the tumor by losing that antigen. And so we are starting to observe clinical responses in this case. CAR T cells will have activity in solid tumors. And there are additional gene editing non-viral approaches that are ahead, so stay tuned. So thank you.
Time for questions. Lisa? Where's the cube?
Other catch.
Yes.
So can you speak to, you mentioned about the antigens. So what would be the best antigen? I mean, we know CD70, most of these CAR T cell therapy trials are targeting CD70, but we heard and we know that there is a trial targeting ENPP3. Do you believe then, what you're advocating, combining, targeting two antigens, let's say carbonic anhydrase 9 and CD70 or CD70 and ENPP3, that strategy. And then again, maybe we can talk about this strategy with cellular therapy later at the end, but I didn't see any of the talks talking about or offering the CAR NK approach of cellular therapies. So two questions, appreciate that.
Bispecific CARs are being clinically evaluated in hematopoietic malignancies now. And so the potential of a CAR that targets CA9 and CD70 are there. I'm sure there are some that are preclinically developed. There's an approach that MassGen is running clinically now in glioblastoma, in which you have a CAR that targets one antigen and then that same CAR T cell secretes a bispecific antibody against a second antigen. So you engage endogenous unmodified T cells to target a second antigen. I think that's an approach that may work as well. One of the limitations of having normal tissue expression of CA9 can be overcome by using a Boolean logic gated system, such as the SYN-Notch molecule, in which identification of CA9 turns on expression of another CAR, or identification of CD70 turns on expression of CA9. And CAR NK cells are in clinical development now. They have not shown similar levels of persistence to T cells. They are off the shelf initially, and so it's a good at least cell source for this type of work. Yeah, they're still coming.
So two questions. One is sarcoma, especially some of the more pediatric sarcoma. There's a high activity of CAR T cells in that group of patients, from what I've seen, more than any other solid tumors. Do you have any idea why that? And the second thing is, some people believe that CAR T cells will be out of business in a little while when bispecific antibodies get improved and you can target CD3 and whatever else you want. And I guess at Penn, you can't look at that, but
No, we can't.
how does Carl think about it?
Yeah. Sorry, your first question was about sarcomas. I actually don't know much about CAR T cell activity in sarcomas. The best activity we've seen to date has been targeting GD2 and DIPG in pediatrics. And that's a very promising trial that's running at Stanford now. There are CAR T cells in sarcoma in development that I know of, but not
Why so?
Oh, but so NY-ESO it's not a CAR. It's a TCR that's HLA restricted. And you're right, those have shown activity in sarcomas by GlaxoSmithKline and Adaptimmune. And then the second question was about bispecifics. So there are several bispecifics that are currently approved now. The first being blinatumomab, which is an anti-CD19 CAR, and blinatumomab doesn't have the same level of activity as tisagenlecleucel or axi-cel, and presumably because these bispecifics aren't adding in co-stimulation to the T cell, aren't allowing for a survival signal in the cells, and stimulation of just CD3 alone can drive T cell energy. But there are improvements that are underway to overcome that, including tri-specific antibodies, et cetera. Gordon.
Question over here and then one from the online.
Wait, over here. I'm sorry. Yeah, hi, Avery. Thank you for that nice review. And I just want to make a comment on the bispecifics and tri-specific antibodies. I mean, they're not living drugs like CAR T cells and CAR NK cells. So there is a difference in terms of longevity of response. Avery, what about payloads? You know, the field is moving towards armored cars and things with payloads in it. Can you comment on that? Because I think that is a distinction, because we do get combination immunotherapy out of the CAR cells if we try that approach.
Yeah, I mean, the inclusion of IL-12, my group is interested in IL-23. There's a trial running now with secretion of IL-18 that's shown, and actually there's another trial in China that counterintuitively is secreting IL-10 and showing better activity of their anti-CD19 CAR T cells. So adding in an additional molecule into your CAR may enhance the potency. Similar to removing some of the inhibitory receptors or molecules may enhance potency of these CAR T cells. I know you had some work presented in a poster yesterday of CAR T cells that secrete a blockade PD-L1 or PD-1, and these approaches, I mean, those are payloads as well that may enhance the activity of not just your CAR T cell, but other endogenous T cells, and then cytokines can do the same for other endogenous immune cells.
Maybe a quick one from the audience. Are there any switch CAR Ts in human trials for RCC right now?
There are not.
So with the non-syngeneic CAR and NK cells, when does anti-cell reactivity develop, and when does it become a problem for eliminating those allogeneic cells?
In the CA9 work, the development of the antibodies was very fast. So antibodies were developed within, I'm going to say, before 2 weeks, and the cellular response was later. This has been observed in others. We've seen this in dogs that we've treated with CAR T cells, developing a canine anti-mouse antibodies, and in humans, repeated injection of an mRNA-based CAR with a mouse SCFV has also led to anaphylaxis in patients. And so we think that that's a limitation for CAR T cell activity because it limits their persistence.
Thank you very much, Dr. Posey. Fantastic summary.
For CAR T-cell therapy in renal cell carcinoma. Our next speaker is Matthew Dallos from Memorial Sloan Kettering. He will talk to us about interleukin-1 beta targeting to overcome resistance.
Great, and thank you for having me and for the invitation. And I'd like to start. This is part of a translational science award from DoD and KCRP, and so I'd like to thank the funders up front and KidneyCAN for really advocating for these sort of translational partnerships. And this is a partnership between myself on the clinical translational side and Michael Shen from Columbia on the basic science side. So I'm going to talk about how we're thinking about targeting the myeloid compartment in RCC, specifically targeting a cytokine interleukin-1 beta. And I think this has been discussed sort of exhaustively in this conference, and I think highlights the challenge of how do we build on immune checkpoint blockade plus or minus TKI. And in the neoadjuvant setting, there's currently no role for IO, but I think that this is really our opportunity for cure in the highest percentage of patients. And so we need to develop novel approaches to this. And I think on the biological side, it gives us an opportunity to understand potentially mechanisms of action and really do the deep dive into the tumor microenvironment. And then immunologically, there's some reasons to think that neoadjuvant therapy may be more effective. You have, when the primary tumor is in place, you have tumor infiltrating lymphocytes present. You have an abundance of tumor antigens, as well as the tumor draining lymph nodes, which provide a site for cross-priming. And then recirculation of reinvigorated T lymphocytes that can then go and eliminate sites of micrometastatic disease. And a shift when you remove the tumor to more of a memory phenotype. And so our interest in the myeloid compartment in RCC really builds on this work from my mentor, Chuck Drake, as well as two fantastic collaborators, Andrea Califano and Alex Obradovic on the computational side. And what they did is they profiled RCC tumor, localized RCC tumors in both tumor and adjacent normal with a CD45 sort. And then they used a computational approach which infers protein activity using an approach developed by the Califano lab called Viper. And what they found is that in RCC, there are really four sort of clusters using this approach of macrophages. And there was one group of macrophages that had high TREM2 expression, high ApoE and C1q expression, which in the exploratory cohort was associated with poor outcome, as well as in a validation cohort. And so how can we think about targeting the myeloid compartment? Why are we interested in IL-1 beta as a cytokine? Well, a couple of points. The first is that IL-1 beta is an innate cytokine. It's polyfunctional, but it plays a role in recruitment of granulocytic MDSCs into the tumor microenvironment, as well as it can drive repolarization of macrophages toward a sort of M2-like state. We also have some clinical data on IL-1 beta targeting. So there's a drug called canakinumab, which I'll tell you more about. But really the basis of this actually, canakinumab was tested in a 10,000 patient trial in cardiovascular disease. And they showed that there was a decreased incidence of cancer across the board and it was a dose-dependent effect. And so based on these insights, David Agin, who's a good friend and collaborator in the lab, tested anti-PD-1 alone, anti-PD-1 plus IL-1 beta and IL-1 beta alone versus vehicle in the Renka model and showed decreased tumor growth. And then when he characterized the immune infiltrate, there was very little modulation of the T cell compartment. But in the myeloid compartment, we saw a fairly dramatic decrease in the granulocytic MDSCs, as well as a shift toward more M1-like phenotype. And so based on this work, we designed a study we call the SPARK-1 trial, which was a feasibility study of 14 patients, so small, but the goal really was to understand biologically what's happening in this setting. And so patients received 2 doses of therapy starting 6 weeks prior to surgery with the combination of spartalizumab, which is an anti-PD-1 drug, and canakinumab, which is a drug that, as I mentioned, targets IL-1 beta. And so I'll show you a little bit of the preliminary. So we've enrolled 11 patients to this study. 9 have gone to surgery. 2 are still on treatment at the moment. This is the sort of baseline characteristics. You can see this is a high-risk population. Most of these tumors were T3. And if you put them into the MSK nomogram at 5 years, the recurrence risk would be predicted to be over 60%. And so far, now this is a huge grain of salt, but so far we have seen no recurrences. And one of the advantages of doing this study, I guess pre-COVID, starting it before COVID and then having a long break, was that we have some reasonable follow-up in these patients. And so we have 4 of the 9 patients are now at 2 years, and a couple of them now up to 3 years follow-up with no recurrence. And so to sort of further interrogate the effects in the tumor microenvironment, we took a broad approach using single-cell RNA-seq, both with the CD45 sort and the total. We developed a flow cytometry panel, which has both myeloid and T-cell markers. For the spatial aspects, we're using multiplex immunofluorescence. And then Dr. Shen's lab is developing organoids with T-cell co-culture so that we can potentially build on this strategy. And these are the core members of our team: Carrie Runcie, who's a clinical investigator; Casey Yeager, who's was a postdoc and now has his own lab at the Mayo in immunology; and then Alex and Andrea from the bioinformatics side, and Michael and Alana from the organoid side. And so I'll show you a little bit of data that we have so far from these patients. So this is from the flow data, really showing that we see a shift in the macrophage populations toward a more M1-like versus M2-like phenotype with treatment. And then looking at the single cell data, actually overall, the myeloid infiltrate increases with therapy, but those TREM2 macrophages that I mentioned earlier are dramatically decreased with this therapy. So we're excited about the biological activity. And so, bringing this back to, where do we kind of go from here? From the bioinformatics side, Andrea and Alex are planning to analyze the data, look for potential novel targets using master regulator analysis and sort of the oncotarget pipeline that they've developed, and then utilize their OncoTreat to potentially identify combination strategies. And then we're going to use the organoids to test this in the culture system. And I think the first step of that aspect of the project is really just to establish the organoids. And so this is a picture of the organoids, both the normal renal parenchyma, the RCC, a case, an untreated case. And then actually see with the combination, it's a little hard to see here, but we do see increased probably tumor killing from organoids developed from the patients who received the combination therapy. So some evidence of sort of functional activity. So in summary, we think that, adaptive myeloid resistance is a key mechanism of immune escape for RCC. We think IL-1 beta is an important upstream cytokine that promotes this phenotype. And we can potentially target it with a drug that's very well safe and well tolerated. And future studies will look at the functional aspect and potentially look at further combination therapy. So with that, I'll thank our group, the participants who signed up for our trial, many of the people I mentioned along the way, our funders, and all of you for your attention. So thanks.
We have a few minutes for questions. Is that? We need the cue.
So great talk. Just wanted to ask if you believe targeting the IL-1 pathway is important. Wouldn't it be better to block both alpha and beta rather than just the beta?
So I think that most of the sort of the suppressive phenotype is actually coming from the beta versus the alpha. So, there's obviously the other drug that's been around for a while is anakinra, which is sort of a decoy of the IL-1 receptor A and binds to both IL-1 beta and alpha. So one could think about that as an alternative drug, but I think beta is probably in this context more important for driving in the tumor the immunosuppressive phenotype.
There's a question over there.
Great talk. Follow-up question on this. The clinical data now with bevacizumab and canakinumab in terms of using this as a therapeutic are interesting. Any comments on that with regards to how this influences your thinking?
That's a great question. One of the things I didn't mention is the challenge with this drug is so the CANTOS trial, which is the cardiovascular disease study, they showed that actually it was lung cancer where they saw the biggest decrease in incidence. And so actually 3 phase 3 studies were launched in lung cancer for canakinumab, and all 3 have read out as negative. And so I think that poses challenges. But I think what these data show, and I think what we've heard over the last few days is that the myeloid infiltrating in RCC is critical and maybe not as much in lung cancer. And so I think it still is worthy of development, but I think it introduces challenges because of those negative studies with canakinumab.
So certainly huge setback with all these negative study in lung cancer, dampened the enthusiasm. Do you know if Novartis is trying to look at subgroups that could benefit? And do you think we should have done that earlier or for prevention? Because there is nice data from Charlie Swanton, with pollution and all this and IL-1 beta, playing a role. Did we go too far into metastatic incurable disease?
It's a good question. So one of the canakinumab studies, there was a phase 2 looking in lung cancer in the neoadjuvant setting. And it does look like similar to what we see is that you can reprogram the myeloid compartment with IL-1 beta. That's the data we have so far they haven't sort of published all of that data yet, but the clinical data in more advanced disease and metastatic disease would suggest, that sort of a lack of efficacy. Now the biomarker work, circulating, I'm not sure circulating IL-1 beta maybe matters as much as what's happening in the tumor, and it's fairly low in RCC, and also I didn't show it, but in our hands, so variable, but sometimes not even detectable with your routine, ELISA assays. So, but whether CRP or other things are predictive are worth looking at as well. Yeah.
Maybe one last question over here, and then we'll.
Yeah, so it's very nice that when you looked in the tumors, it kind of replicated what you saw the mice decrease MDSCs. But kind of relevant to all these questions, I was curious, do you think that it's the MDSCs, for example, in cardiovascular patients that prevent them from getting cancer? And more importantly to you, do you have ideas? Are you banking on that it's the MDSC mechanism that's going to, prevent recurrence of disease in these NED patients? Or have you thought of other mechanisms that IL-1 might be doing those 2 things?
Yeah, so I probably can't speak too much to the cardiovascular aspects of it, but certainly from our perspective, the MDSCs I think are critical. I didn't show it here, but the same sort of you do see a depletion in the.
Is that in that trial, which was a cardiovascular trial,
Yep.
It was prevention of cancer,
Right.
right? And so do you think in the prevention of cancer or the prevention of recurrence of disease, do you think the MDSCs are playing a role? I mean, clearly you showed in the tumor, it was nice. You showed the effect on the MDSCs, but I was just curious about that.
Yeah,
aspect.
I mean, yes, thank you for clarifying the question. Yeah, I mean, I think it's an important thing to consider. I mean, I think in RCC, we think that as you get more, and David has shown really nice work on this as well, as you get more aggressive disease and progression of disease, you get more, MDSCs. So whether or not, early as a prevention, that's how we want to go. I'm not sure that I would biologically think that that's the case, but maybe you don't need, these are very immunosuppressive. The granulocytic MDSCs are very immunosuppressive, and maybe you don't need that many in the milieu to help, trigger sort of tumorigenesis there. So I don't know, but we do think of MDSCs as being something that develops with tumor progression, and the infiltrate gets more significant. So
Thank you so
all
much.
right, thanks.
That's great. Next, we have Dr. John Wilson from Vanderbilt talking about enhancing RNA sensing in kidney cancer.
Okay, thanks everyone. I'd like to thank the organizers for having me here. I'm John Wilson. I'm actually in the Department of Chemical Engineering at Vanderbilt. So I'm a bit of an outsider, and I thank the community for welcoming me during this meeting. It's really been a great time to learn a lot. So today I'm going to talk about some of our work in harnessing RNA sensing, particularly the RIG-I RNA sensing pathway to enhance responses to immunotherapy cancer. Forward. What's forward? So now, can I get it? Do not press. Here we go. No, that's the other wrong way.
The green one.
Oh, there it is, a big one. Okay, nice. I'm colorblind. It's a good excuse, right? All right. So, okay, so we're all here for a common reason. We all understand this challenge of immune checkpoint inhibition. Inhibitors have revolutionized treatment of kidney cancer, but there's still work to do. And this is what really this session and a lot of this meeting has been about. And so the challenge that we and many others in this room think about is how can we overcome these less immunogenic tumor microenvironments and tumors in order to ultimately improve responses to immunotherapy in kidney cancer. And so that's it's one of the challenges that my lab works in. We do this in a number of cancer types. And so again, we are engineers. I consider myself a molecular engineer, so maybe not working on ships or boats, but working on or trains, but instead working on molecules, engineering molecules of various length scales and sort of molecular compositions. And so we're sort of trying to work at this interface of engineering and immunology, and with the goal of ultimately developing new strategies to improve responses to immunotherapy. And so we work in areas of innate immunity, which I'll talk about a lot today, cancer vaccines, which we heard some about earlier, and as well as some other types of modalities for immunotherapy. So again, what I'm talking about today is our interest in innate immunity. I think most people in this room can appreciate that innate immunity is involved in virtually every phase of the cancer immunity cycle, everything from antigen recognition to MHC1 presentation to cross-priming, et cetera, T-cell infiltration. And so if one can sort of pharmacologically manipulate innate immunity, you have opportunities to intervene within multiple steps of the cancer immunity cycle with the potential to make an impact in responses to immunotherapy. And so we've worked in a number of different innate immune pathways. We've done a lot of work in CGAS and STING signaling. We've heard about that some already. But what I want to talk today is some of our work in RNA sensing. And so in particular, I want to talk about the RNA sensor RIG-I. And so RIG-I is a cytosolic pattern recognition receptor. It's involved in recognition of several types of viruses. It recognizes double-stranded RNA with a 5' or 5' triphosphate or diphosphate group that is not associated with typical normal healthy RNA. And upon binding of double-stranded RNA to RIG-I, it initiates a type 1 interferon-driven antiviral type innate immune response that can be correlate or can be leveraged to enhance T-cell infiltration and function in the tumor microenvironment. So our interest in activating RIG-I in the context of kidney cancer was really inspired by work from several labs, including Kim Rathmel's lab at Vanderbilt, who show that activation of endogenous retroviruses can generate a sort of antiviral like cytokine signature in the in tumors, and that this might correlate better with responses to immunotherapy. So just a quick, some evidence of increased RIG-I expression, DDX58 RIG-I correlating with survival, and then data from one of Dr. Rathmel's paper, Kim's paper, showing an increased expression of RIG-I in patients that respond to anti-PD-1 or anti-PD-1 immunotherapy, sort of motivating us to look into this pathway. So again, the objective of our project is to develop and optimize a platform for pharmacological activation of RIG-I. So 5' triphosphate and diphosphate RNA are they're very potent ligands for RIG-I, but they're bad drugs. They have a lot of the same problems that all oligonucleotide therapeutics have, as well as mRNA. So they need to be packaged, they need to be protected, and they need to be ultimately delivered to the cytosol where they can then be released and bind the target of RIG-I within the cytosol. And so our lab works on this a lot. We have a lot of different sort of strategies for packaging nucleic acids, getting them to be more efficiently delivered to the cytoplasm. But in this project, we started pretty simple. We said we want to use something that is fairly already translationally advanced. So we picked a lipid nanoparticle. And so this is lipid nanoparticle. Our first design iteration is based off of a lipid nanoparticle that's already in an approved therapeutic for siRNA. And again, most of us are now familiar with lipid nanoparticles. We've all, at least the majority of us, have probably had them injected into our arms a few times. And so we can then take the RIG-I agonist here. We're using a 5' triphosphate RNA that was developed at Yale in Anna Pyle's lab. Anna Pyle and I continue to collaborate together to develop new more defined and more potent RIG-I agonist. So we took a RIG-I agonist from her group, we packaged it into a nanoparticle, made some tweaks to that. And as a result, we can get a strong interferon response to that particle. C, in this case, you'll see the C in front of the CLR there is a control RNA that lacks the 5' triphosphate group. So but the data I'll show you now is going to be in melanoma, and I'll end with a little bit of preliminary data in renal cell. So this is data in melanoma, first looking at the RIG-I agonist nanoparticle in the context of an intralesional therapy. So again, B16 melanoma with 2 tumors, inject 1 tumor, again, also treat with immune checkpoint inhibitors. And you can see here that we get both control of the treated tumor as well as the untreated distal tumor. So suggesting an abscopal effect that's mediated by this nanoparticle-based RIG-I agonist. Importantly, where we want to push this is to be able to use this as a systemic therapy. And so we have some evidence that we can use this systemically right now in its current form. And this is, again, a melanoma model. This is a lung metastatic model of B16 melanoma, where we inject into the tail vein, and then we treat with the nanoparticle or checkpoint inhibitors. And what you can appreciate here is that the nanoparticle RIG-I agonist, but not the control nanoparticle that lacks the 5' triphosphate groups, gives us a fairly, a strong response in this lung metastasis model that is then further enhanced through addition of the immune checkpoint inhibitors. So finally, this is our early stage data, and this is, again, what we want to build upon, is some evidence that we can do this in a model of renal cell carcinoma. Here using the RINCA model, where we again establish the RINCA model, is treat with the nanoparticle RIG-I agonist 3 times, and we can see that we do get reduction in tumor growth as well as some extension of survival in the mice. So at least providing some initial evidence that this is a viable target for treating renal cell carcinoma that will allow us to sort of move forward in our design iterations and in sort of further testing and expanding this therapy. So again, sort of the major project goals for this, related to this project, we want to further improve upon this nanoparticle based approach for RIG-I agonist delivery, both through the nanoparticle design as well as through the agonist design. Again, we started pretty simple. This is far from optimized for systemic administration. So that's one of the big goals, is to improve upon that. Then to understand the immunobiology about what happens when we activate RIG-I in the renal cell tumor microenvironment. And this is work that's being done in collaboration with Jeff Rathmel, Kim Rathmel, Katie Beckerman, and being supported by our really great patient advocate, Tammy. So with that, I'd just like to thank everyone again for their time, for inviting me here, as well as to my research lab. So with that, I'm happy to take questions.
Questions?
From there.
Eric?
Great talk. A question about the stability of this particular molecule and whether or not a nanoparticle is the only way that this can be delivered, and are there alternates? I'm just sort of thinking, we have great cell surface targets, and, are there ways to deliver this more precisely?
Yeah, that's a great question. I think the answer is yes, but there's some ways to go in the development. I think what it's going to take is the design of a very chemically stabilized RIG I agonist. And the biggest liability, I think, is the triphosphate and diphosphate group in terms of the stability of those bonds. There's a lot of really good medicinal chemistry that's been done in siRNA therapeutics that allow those to be much more stable, sort of get away from some of the nanoparticle-based delivery. So that is probably, long-term where the future would be.
Thanks.
Thanks over there to Wayne.
That was a total fail. If anybody saw that. I hope you guys have insurance for personal injury with this device. A follow-up to the last question, and one that David Braun brought up, which is targetability. So my understanding is that lipid nanoparticles really tend to have non-specific uptake into the liver, for example.
Yeah.
So are they, is the technology to the point where they are targetable? I mean, if we have a ligand that is specific for RCC, is there enough progress in the field to that non-specific uptake can be overcome?
It can be reduced, but not overcome. Targeting will give increased accumulation at tumor sites, but it's still a limitation. You will have clearance through other organs of nanoparticles. There's a couple ways that we're trying to kind of, I think, work around that. One is, I think, we've found that for other innate immune agonists that having a appropriately tuned half-life is important. And so trying to get half-life extension of the particles is one. You can route them to, for example, the spleen instead of the liver. And I think there could be immunological advantages of doing that to sort of mobilize cell populations of the spleen, or perhaps get antigen priming of the spleen, those sort of things. So that, getting them mostly off the liver and into the spleen is a fairly, that can be done. The other thing that we've done a little bit is, and sort of to the discussion of, can this go beyond a nanoparticle in other formats, is we've done some early work, sort of asking, can we convert the RNA itself into more of a prodrug like molecule? So even if it does accumulate in other organ sites, that it might not be active.
Other questions? Okay, if not, then let's move on to the next speaker. Thank you so much. The next speaker is Marice Alcantara from the City of Hope, speaking about a novel STAT3 antisense oligonucleotide-based immunotherapy for renal cell carcinoma. Thank you so much.
Hello, everyone. Thank you for allowing me to speak with you all today. As mentioned, my name is Marice, and I'm in the Kordaluski lab at City of Hope. Today, I'll be talking to you about a novel STAT3 antisense oligonucleotide immunotherapy for renal cell carcinoma. So the interactions between tumor cells and immune cells in a tumor microenvironment is highly complex. And we know that the inhibitory pathways used by the immune system to maintain tolerance is often hijacked by tumor cells. We know that immune checkpoint blockade targeting these inhibitory pathways have improved responses in renal cell carcinoma patients, but we know that a lot of patients do not respond, potentially as a result of significant immune suppression that's coming from outside the T cell compartment that's being targeted. Now, in a recent study that was shown by Disman and colleagues, they showed that the gut microbiome and the modulation of the gut microbiome can enhance responses to combination anti-PD-1 CTLA-4 in RCC patients. And this is potentially the result of innate immune cell activity. Indeed, innate immune cells, such as myeloid cells and macrophages, are known to be regulators of immunosuppression in many solid tumors, including renal cell carcinoma. With myeloid cells demonstrated critical components of resistance to immune checkpoint blockade, as the people who presented before me have discussed. So we postulate in our group that targeting myeloid cells can improve patient immune responses to immune checkpoint blockade. Now, the tumor-mediated hijacking of myeloid cells depend on STAT3. It's a transcription factor that's activated by various cytokines and activated molecules, including IL-6. Now, STAT3 deletion in myeloid cells has been shown to result in enhanced anti-tumor cytotoxic T cell responses and decrease the suppressive T-regulatory cells within many models. And in renal cell carcinoma, STAT3 deletion actually STAT3 actually correlates with RCC growth and a worse prognosis. And when you combine this with the reported role of myeloid cells in immune checkpoint blockade resistance, this suggests the potential for myeloid cell-specific STAT3 targeting in the disease. So this study was actually done in two parts. The initial part was doing some immune monitoring in patients who received combination anti-PD-1, anti-CTLA-4 therapy in 2 clinical trials at City of Hope. 37 patients who were enrolled and who received first-line therapy anti-PD-1 CTLA-4 were used. The patient characteristics are listed below. We then looked at various immune cells and cytokines circulating with these patients and stratified them based on responders and non-responders. What we found was that there were various cytokines that were elevated in both responders and non-responders to therapy. Notably, IL-6 and IL-8 were only significantly upregulated in patients who did not respond to therapy. IL-6 and IL-8 have a role in STAT3 activation and myeloid cell recruitment, suggesting myeloid and STAT3 signaling in non-responders to immune checkpoint blockade. In addition, we also observed that within the immune cells, looking at activation of STAT3, there was evidence for STAT3 signaling within the CD33 positive where myeloid cells are contained in patients, and this is before treatment at baseline and then after week 12. So it's STAT3 signaling before treatment that's maintained throughout treatment. And so we wanted to investigate if STAT3 targeting in tumor-associated myeloid cells might have any potential in the disease. And we utilized the drug that's been developed in our lab, CpG STAT3 Antisense. It's a STAT3 inhibitor that's conjugated to CpG. This facilitates two things. So the CpG portion allows for specific internalization by myeloid cells. And then the STAT3 antisense allows for specific RNase H-mediated cleavage of the STAT3 mRNA. And this results in two things. The reprogramming of myeloid cells, which then aims to support T cell mediated anti-tumor activity. In other models, such as prostate cancer models, our group has shown that it improves survival in prostate cancer and also decreases tumor cell growth. So I then proceeded to test anti-PD-1 with CpG STAT3 antisense in the Renka mouse model delivered systemically and showed that there was, in combination treated animals, there was a decrease in tumor growth kinetics when you compare them to controls that are PBS treated or IgG antibody treated control, and as well as the anti-PD-1 alone treated animals. I'm going to focus primarily on the myeloid cell compartment in the interest of time. What we found was that most of the significant changes were within the myeloid cell compartment, where there was a shift from the M2 phenotype that's evident in the controls, PBS, the IgG, and even the PD-1 alone treated groups to a more M1 phenotype with combination treatment and CpG STAT3 antisense. But really what we want to focus on is reprogram these myeloid cells to support T cell function. And what we found was that combination treatment can enhance CD8 positive T cell infiltration within tumors. Now we know that CD8 positive T cell infiltration doesn't generally correlate with improved responses. And we see that there is CD8 positive T cell infiltration already within controls, and it is comparable to PD-1 blockade alone, but we can further increase that with combination treatment. And then when we look at the ratios between CD8 positive T cells anti-regulatory cells, we see that we can further increase the ratios, increase the CD8 positive infiltration in comparison to the suppressive T regulatory cells if we use combination treatment, suggesting that reprogramming myeloid cells may enhance T cell function in the disease. So finally, I would like to thank everyone who contributed to this work, my mentors, Martin Kotolewski and Dr. Samantha Paul, and the labs in Kotolewski lab, the Swiderski lab, and also everyone in the clinical team, our clinical collaborators, and the CDMRP that allows me to do this work. Thank you.
Questions? Let's start off with,
Let's see.
Thank you, great presentation. Part of me does melanoma after hours. So a couple of years ago, we published on CPG in a refractory setting using TLR agonist in PD-1 inhibitor. And we have seen very, very interesting results. It's published in Cancer Discovery. So
I would love to talk to you more.
Yeah, that would be great. So what are your efforts? Are you doing anything in kidney cancer prospectively in clinical trial setting?
So we're at the very beginning stages of this research. We've only been funded for a year, but that's definitely something we would like to look into. And we're letting the results guide us, but definitely. And if that's okay, I would love to chat to you more about your research as well.
Anyone else?
I'm going to ask a quick question as well, which is, it seems like you're targeting sort of the downstream STAT3, but sort of upstream of that, there's IL-6 and IL-8, and maybe just a couple comments on advantages or disadvantages of targeting IL-6, IL-8 upstream versus STAT3 directly.
Okay, cytokines have so many varied roles in the disease, and we want to focus on primarily activating the immune system and supporting the superstars of the immune response, which is T cells. But of course, we know that T cells, any superstar, requires a huge team, right? Michael Jordan didn't do everything on his own, despite what he says. So that would be my answer to it. I mean, there would be potential for that, but I don't think the cytokines themselves would be the main targets. I think it would be completely how do we support the superstar T cells, and how do we look at patients who don't have risk, like who develop resistance and don't respond to combination therapy. Let's look at them, let's support them, guided by the the immune alterations that the resistance is showing us. Thank you.
I have a question for some of the melanoma docs here. So I know that some of these trials, so the theme a little bit has been, maybe some cytokines that are detrimental to the immune response or immune suppressive, et cetera. I know there were some trials going on with anti-TNF in combination with immune checkpoints in melanoma, potentially enhancing the immune response while suppressing toxicity. Any experience from the melanoma docs here that can give us some decision? Thinking along the lines of like IL-6 inhibition, IL-8 inhibition, anything. I know I see some melanoma docs here. Okay, good. Please, yes.
Yes. So, hello. Yeah. All right. Good. So yeah, we've done some work with IL-8 antibody as well, more in the context of prostate cancer than RCC, which is a much colder tumor. I think the long story short is when you look at cytokine levels, when you block IL-8, actually IL-1 beta goes up. So I do think there's a sort of a cascade of which one is sort of at the top of the food chain, which one's more upstream. I think IL-1 is upstream of IL-8 at least. IL-6 is one of the other innate cytokines but that doesn't go up quite as much when you block IL-8. So I mean, I think these are interesting targets, but you probably will need to ultimately hit multiple cytokines or something downstream as you guys are considering.
Great.
Yeah. It would be also interesting if we could like utilize these cytokines as like a biomarker of patients that won't respond to therapy. So like that's something we're looking at in the group. Thank you.
Great, thank you so much.
And then our final speaker for this session is Dr. Qinqin Jiang from Dana-Farber, who's going to speak on endogenous retroviruses in kidney cancer.
So I'm a postdoc fellow in the Cable Lab, and thanks for KCRS for giving me the opportunity to present here today. And our research aims to identify HIF-regulated endogenous retrovirus in CCRCC, and hopefully we can provide some implications for immunotherapy. So CCRCCs are known as immunogenic tumor due to several clinical observations, including that tumor occasionally regress spontaneously and high level T cell infiltration often observed on the tumor pathology. And it's been responsive to immune modulators to various degrees, such as IL-2, interferon alpha, and in more recent year, immunocheckpoint blockade. Normally, good response to immune checkpoint blockade correlates with high mutational burden. And CCRCC in that perspective is kind of an outlier where it has a fairly good response despite modest mutational and neoantigen level. So that leads us to think that whether there's a CCRCC specific tumor antigen that causes immunogenicity and it's just not yet been discovered. So one category of such antigen could be from endogenous retroviruses. So in short, ERVs. So what are ERVs? They're remnants of exogenous retroviruses that integrated in the primate genome millions of years ago. Structure-wise, it's very similar to exogenous retroviruses and is highly abundant in the genome, comprised about 8% of the human genome. Due to negative selection during evolution, these ERVs are largely silenced either by mutationally or epigenetic regulation. Nonetheless, some of the ERVs still maintain the open reading frame and have the potential to be translated. So in the CCRCC context, previously been shown that ERV appears to be upregulated and they're associated with immune signatures. And in some clinical CCRCC cohorts, we have observed that expression of ERVs correlate with improved immune checkpoint blockade response. Since aberrant HIF signaling is such a key feature in CCRCC, so we wonder whether HIF could lead to immunogenic ERV antigen expression on the cell surface that lead to the immunogenicity. So Richard Child's group discovered one such ERV, which expression is driven by HIF and is immunogenic. What it means is that they actually discovered in patients circulating blood T cell, in patients circulating T cells, they could recognize ERV-derived peptide and lead to tumor regression. So our overarching question is that since HIF2 is such a master transcription regulator in CCRCC, and their ERVs are very abundant in the human genome. So we wonder whether ERV4 was just one example, and then there are additional HIF-regulated ERV at transcription level, maybe at translational level, and eventually these HIF-responsive ERVs are being translated and presented by the HLA complex cells on the cell surface to invoke and to lead to immunogenicity of CCRCC. So first, we want to identify HIF-responsive ERV systematically and unbiasedly in the genome. So we created three comparisons where HIF activity is high and low, as shown here. And then we're trying to evaluate the abundance of over 3,000 annotated ERV in the genome. So here we can see that with three comparisons, 16 of them scored as HIF-dependent ERVs, and reassuringly, ERV4 was one of them, suggesting our pipeline is probably robust and working well. So next, we're wondering whether HIF2 is actually localizing to these ERV sites. So we did a HIF chip-seq in the CCRCC cell line, and we identified over 23,000 HIF2 binding sites. And for 105 ERVs, they do have a superimposed HIF2 binding site. And for 371 ERVs, they have a HIF binding site within 10 kilobase. And when we overlap this data set with what we identified in the previous slide, the HIF responsive ERV based on RNA-seq 10 of them overlap. So to ensure the robustness of this system, we knocked in a flag tag on the N-terminus of HIF2 at the endogenous locus and did flagship seek to look for HIF2 location in various cell lines. And below are examples of two ERVs where we can see that in using different antibodies as well as in different CCRCC cell lines, consistently HIF2 is localizing to the ERV sites. So next, we're trying to understand whether any of these ERVs are actually being translated. That's the ones that before they're actually being presented on the cell surface. So we did a polysome seq in high HIF and low HIF status in the CCRCC cell line again. And the translated RNA that associated polysome were sent for sequencing. And then we could see that on the right side, we identified 26 ERVs that are regulated by HIF at translational level. And when we overlap that with RNA-seq, we can see 4 of them, including ERV4. So now we know that there are ERV being regulated by HIF at transcription and translational level. The next step is whether any peptides presented by HLA is actually in the HIF-dependent manner. So we use our isogenic cell line where they express either empty vector or VHL, and then we pull down using a class 1 HLA complex and elute the peptides that bond to the complex and mix them after chemical labeling these peptides and mix them together, which allows us to do quantitative mass spec. So on the right is a volcano plot. Each dot represents a peptide identified in the mass spec. So these peptide could be derived from just open coding genes or new ORFs or HIF-regulated ERVs. So to test the robustness of our system, you could see that HIF target gene and DRG1 derived peptide is higher abundance in high HIF status, and VHL derived peptide is more abundant in low HIF status, suggesting this quantification is reliable. And there are 2 ERV derived peptide are being identified as HIF responsive. So from the cell lines, we learned there are HIF-regulated ERV at ERV as well as peptide. So to test the immunogenicity of these ERV-derived peptides, we collaborate with Dr. David Braun as well as Kathy Wu, because they have clinical samples where in their Neovax trial, they have done mass spec on 11 tumor samples, as well as 6 matching normal samples to identify HLA bond peptides. So looking through that database, we were able to identify 34 ERV-derived peptides. So these ERV-derived peptides were from HIF-responsive ERVs, and they are tumor-specific when paired normal tissue samples are available. So currently, we're in the process of determining the immunogenicity of these 34 peptides using these corresponding patients' PBMCs and TILs, and also humanized mouse. So we hope that our study could open doors to some potential immunotherapy development, such as using CAR T cells to recognize ERV-derived peptides, or these peptides could be good cancer vaccine, especially consider these sequences are from viral background. So with that, I would like to thank my mentor, Dr. Kalin, and all my collaborators over the years and the funding from DoD. Thank you.
Eric has a question. Let's see him catch.
Great talk, very interesting. So based on your findings, what would you hypothesize would happen if you use HIF-2 alpha inhibition together with immunotherapy?
Yeah, that's a very fair question. And then we discussed a lot with Bill and with other collaborators. I think if we think all HIF does is really just to the cancer cells, and then in my mind, maybe immunotherapy would be a first-line therapy to first harness that activity, and then whatever is not treated, we can try to use HIF-2. But we don't know how HIF-2 inhibitor is working on the microenvironment, the immune cells, and so it's hard to know overall what the outcome will be like.
I mean, what you're showing is you might actually decrease the immunogenicity of the cancer cells themselves.
Yes.
So
that's why I'm
obviously-
suggesting maybe immunotherapy itself first. And then if that cured the cancer, that's awesome. But if it's not, maybe in the second line or later on.
So it may be a great thing for us to study what these HIF2 alpha inhibitors are doing to the tumor microenvironment in humans, so we can really understand their role.
Yes, I agree.
Tony?
Yeah, I mean, I tend to differ here from you, Eric. I think here, if we assume that the effect is only synergistic, HIF2 inhibitor will be able to killing tumor on their own, irrespective. I think, there is, mouse model from Bob Kerbel, if you remember, with short duration of VEGF TKI accelerating tumor growth in an adjuvant setting, which we show that doesn't happen in a human. We don't have worse outcome. So I think it's extremely reasonable still to combine them, despite, if we have, something. So I'm fine totally with that.
Also,
I think, empirical experiments are fine, but actually backing it up with some kind of a mechanistic understanding of what we're dealing with is critically important. Otherwise, we have clinical observations absent any sort of mechanism. And so that's basically what I'm asking for, Tony.
I mean, yeah, that is fine, but I think the message here that shouldn't, get out and be mainstream is that HIF2 inhibitor are going to decrease the immunogenicity or any, any benefit effect that an immune checkpoint inhibitor is going to have. I mean, all the trials that we're doing and now are in late stage, they had all DMCs, approval after 6 months. I think this will be important to see, not just the interaction of the immune system, but what is the ERV doing there. So, and hopefully we'll proceed better. And if the results are not going, the right way, we have to know how to sequence things better.
Jeff?
Yeah, there was an attempt way back, Steve Balin, and I know there's others who believe histone deacetylase inhibitors actually enhance expression of ERVs and is part of the reason why they enhance the immune response. It hasn't exactly panned out, but I'm sure the ERVs are increased. Have you looked at other mechanisms or strategies to increase their expression?
Yeah, I mean, most of these ERVs are silenced by DNA methylation. So DMNT inhibitor certainly works very potently for a large amount of ERV, and at the same time, that will increase the innate signaling of sensing these ERVs as well. So that's definitely a good candidate. But I think there are also other studies where they're trying to use demethylase and to increase, because basically to open up the chromatin, so that will allow these ERV to be transcribed and maybe presented as well.
Abhishek?
So Qinqin, just a very quick comment in line with what you just heard, but basically there are probably HIF dependent but independent mechanisms to just open up the chromatin in the VHF deficient cells, right? And so it's likely that even if you shut off HIF, some of the K27 acetylation will still be maintained and the ERVs will continue to remain expressed, especially in an in vivo setting. So it would be important to answer the Eric's question experimentally, but I think there is hope to expect that there will be some redundancy built in over there. I have a second idea, and that is we are learning a lot about how melanoma and prostate cancer is informing how we should be thinking about RCC responses. But here there might be one example where we should think about how your studies can inform other tumor types as well. So if you think about other HIF2 dependent tumors, GBM, or some other tumors where hypoxia could be driving HIF induction, have you looked at what is happening to ERV-E4 expression in those tumor contexts, and whether peptides from this ERV would now provide a neoantigen in those tumors as well?
Yes, so we actually have tested multiple in different cancer contexts and to basically increase these ERV expression by HIF stabilizer, which is the approved proxyl hydroxylase inhibitor. And in GBM, for example, in the GSC cells, we were able to derepress these ERVs as well, especially when we combine that with a DMNT inhibitor. But in a little slight different model where, because we're lacking syngeneic model for the kidney cancer, we were doing syngeneic model using like ICC, the liver cancer or pancreatic cancer, and we could stabilize HIF and we could see derepression of ERV in the tumor specific manner, because I think the epigenome in the tumor cells are just more open. So I think there is a window to derepress these ERV specifically in tumor cells in other contexts.
Great, thank you so much. I see Brian Lewis patiently waiting there. I know there's food outside and he will probably say a few words. So thank you so much, everyone, for the presentation of this session. Thank you so much.